mniache, msiniulize swali. Sasa tena. I'm not sharing the link first. Toka Madagascar asubuhi. Acha ngani. Nimetoka mara kama mwami. Wanasikia. All right. Now this is what you are explaining. So this is what will happen. So your blood has excess glucose. It will supply to cells. Then kuna na some fluid between the cells. So your cells, this will be hypertonic. Blood now it will be hypertonic. When they are hypertonic, the cells will be hypertonic. Ama will have lower concentration of solids. Glucose wakati tunasema hapo ukiweka glucose, kuna test nyingine unachorewa. Let us do it vizuri. Now you have two setups here. First setup A and B. Here you have what do we call this back to the last one? So this is whisking tubing, and here tuko nayo hivyo. Hapa tumeweka starch kama mahindi, unga, mchanganya na maji. Alafu hapa uweke glucose ile unajua. Ndio unanunua kwa duka glucose. So unaambiwa it was left for 30 minutes. Explain what happened to the whisking tubing in setup B and setup A. Kama unachora love letter kwa mkono na naona haujakaa karibu na the other agenda. Anyway, so yani you should watch hapa, wacha kuangalia mfano. So what will happen? Tulikuwa tunasema hapa this is distilled water. So for this one, what will you say? The whisking tubing increase. Bulge. Nahi? According to Angel, both will water will get inside the whisking tubing. For you? Kwa starch itaingia, hii ndio itaingia. Zote zitaingia. Both. Wisdom. Hakuna yenye itaingia. Hujui. Huna idea. B. So this one has osmotic potential. Nikaweka mchanga. Mchanga haiwezi vuta maji. Ama haina osmotic potential. Haiabsorb maji. Sawa. Hii ni absorb glucose. Starch is not a reducing sugar. So what I was explaining is that now glucose maji itaanza kupotea from the cells, your body cells. So when your body cells lose water, it is dangerous for the cell's survival. And remember, your survival depends now on the cells. If you cut yourself into small pieces, the smallest functional unit is a cell. Umeelewa? Umeelewa why excess glucose is harmful to your body? Let us, I think when I was saying, understanding but you are not giving me feedback. Kuna kitu inaitwa communicating. When you communicate, inakuwa tu I tell you, you give me feedback. Whether you have question 3, which is patient.
Still the formula for calculating mean multiplication of a specimen is
Formula of calculating? Calculating multiplication length. So what's the formula? Tulikuwa tumeandika na hawa. Huh? Yes. So I don't know why you found it difficult. So magnification is equal to length of drawing or length of image over corresponding length of specimen. So, magnification. I think this is magnification equals to length of image stroke drawing over length of corresponding length of the specimen. Corresponding length on specimen. And I gave you a secret to passing sciences, especially biology. You should now be able to distinguish between biology, chemistry, physics, study techniques zake. For biology, you need to study again and again. Ni a theoretical science yenye nataka uexplain. Unaona chemistry? Chemistry nikikwambia acid plus base is equals to salt and water. That one will never change. That is a principle that will never change. But when you are doing biology, you need to understand those specific concepts that you apply everywhere. You have to study concept by concept. So length of drawing or image is equals to corresponding length on specimen. Studying the next question, Nikaro, please read for us the question.
An investigation of setup is shown in the diagram below.
Then you have the diagram.
After that, that substitution are blue, black, while Eagle's solution will change its color. During the physical process of being investigated in the experiment.
Oh, good. Now, this is so now you are given, you are revising at Form 1 cut. So you have this setup where you have so this is iodine solution. My water is going beyond the container. So this is iodine solution. Here there is starch. You know starch test? When you add starch with the iodine, it forms a blue-black coloration. So this is starch. So it's not starch. Starch. So you are told that this setup was left for 30 minutes. for some time. So an investigation was set up as shown in the diagram below. Glass rod, whisking tubing, starch suspension, and iodine solution. After 30 minutes, starch suspension had turned blue-black, while iodine solution, this had turned blue-black. Remember, starch is whitish, kama unga ama ugali, but it had turned blue-black. But this retained its color. Iodine is it purple or is it? Iodine is purple.
Red brown.
Sorry, red.
Red brown.
Red brown. Yeah, it's red brown. Then this one turns blue black. Name the physiological process that is being investigated. You are saying? She's saying osmosis. Who else agrees with osmosis? Okay. It's not osmosis. Mumekata amesema peke yake. So what is it?
Diffusion.
Huh?
Diffusion.
Diffusion. Who else? Wisdom? Osmosis. Peter? Diffusion. Kesha? Osmosis. So, 3 people say osmosis, 3 people say diffusion. So what is the average? So diffusion, why? We are talking about movement of iodine. Listen, iodine moves from here to here kwa starch test, so that ilikuwa blue-black coloration, which means iodine ilikuja hapa. Not water, iodine. So if it is iodine, ni diffusion of osmosis? Osmosis, the movement of only water molecules. So this is diffusion. Kuna whisking tubing. It is selectively permeable. It doesn't mean it is only permeable to water. It is just selectively permeable. So it is selectively starch haikutoka. Either of them could move, but it did not allow. It allows some particles, not all. For example, one alternative whisking tubing is tube, right? In urine, kuna kwanga na not only water, kuna kwanga na salts. So salt are able to pass through that semi-permeable membrane. So they diffuse out. So even whisking tubing, which is similar to that dialysis tube, ama to our kidneys, can be able to pass out salt. Iodine can be able to pass, and water. So, but not all particles. Just selectively permeable. Some particles can move, others cannot. Are we together? Yes. Account for the results observed. We have already said what happened, but you can say. So, the marking scheme inasema diffusion, then inaandika hivi RJ osmosis. RJ ni reject. Ukipata kwa marking scheme ya biology ama kwa biology inaandika reject ama reg. Reject osmosis. Mtu akaandika osmosis wrong. It's diffusion. Haya.
I have a question. You put a show up and then it's in this protein that is being tubing, and then it's what outside. Physiological process again.
Sorry?
Potassium permanent together and don't do this being tubing. Then you can like distill water. Your physiological process.
So anauliza if we have this is water and then the whisking tubing hii, halafu hapa ndani kuna potassium permanganate. Hivi andiko hivyo manganese form. Potassium permanganate iko hapa ndani, and we know its color. Is it blue? And there is distilled water. Then the results ni that this color ina change because hapa tumeambiwa the color of the iodine just remained the same. So in this case it is more likely tuambiwe color changed. Ama the result ni gani? If its color changed, it is still diffusion. That potassium permanganate diffused out of this. But if it is about this one bulging, it is osmosis. If the observation, let's come look at the observation. If the observation is that this tubing will bulge, ama increase in size, it means water got inside. But in this case, the observation will be potassium permanganate color, ama this water became blue, ama the color of potassium permanganate. You can research. Sawa. Kama ni hivyo, wewe unaangalia tu ni nini na move. Just note, osmosis is movement of water molecules only. Diffusion na active transport, movement of any particle. Sawa. Then sasa tofauti ya diffusion na active transport, diffusion high concentration to low. Na opposite ni active transport. Sawa. Haya, so I think umeelewa hiyo. Ni topic ingine lazima iulizwe. Alafu they just twist the question vile umeuliza. We have a question. The next question is nani? Oh, we are talking about, you can say the explanation on it. Whisking is semi-permeable, allowing the smaller molecules. Whisking, tubing, is semi-permeable. We are writing the explanation. We have said its definition, allowing the smaller molecules of iodine to pass across to the starch suspension. Whisking is semi-permeable. Whisking, semi-permeable, allowing the smaller molecules of iodine, allowing the smaller molecules of iodine to pass across to the starch suspension, while the larger starch molecules, these ones are not compatible. Allowing the smaller particles of iodine to move across, to pass across to the starch suspension, while the larger starch molecules cannot cross to the iodine solution. The larger starch molecules cannot cross to the iodine solution. So that's why iodine solution remains the same. Maybe here ni max 3, but if you are to add, ingekuwa ni 3 max. You know, when starch tunaelezea. The next question natujaribu kwenda mbio. Alright, take a question paper. Ndio ndio. Yes, I would like to read the next question. Kesha, it's you. You ready? Are you sure? If you read, you tell your neighbor. What is osmosis? Define the term osmosis.
The process by which water moves through from region of precipitation to a region of low precipitation.
So is the movement, osmosis is the movement of water molecules from a region of high concentration to a region of low concentration through a semi-permeable membrane. So if you don't write through a semi-permeable, I'm across. We are talking about so across a semi-permeable membrane. You must write across. If you don't write that, you will get penalized. Higher. Then,
Between promoters and currents.
What do you think? Peter? Can you read the question, then you answer, Peter has to have no idea.
Yeah.
So Peter doesn't know what heterodons and homodons are. Nutrition in animals iko form 1. Yes, I have. What is heterodons, anyone? Or homodons?
Pterodons and the animals that have four types of incisors, canines, premolars, and molars. Homodons. They have the same teeth. Hakuna difference.
So homodons possess the same kind or type of teeth, while the heterodons possess different types. It is not necessarily equal 4 types, but they just have different. Example of a homodont. Shark, mashark. Shark ni kama samaki. So you have iko hivi. What about crocodile? Crocodile ikichorwa naona inafanana. Inakuanga na kama ya mwanadamu ama ya mtu. Ndio iko. So homodons have this kind of teeth. Meno inafanana tu yote. Lakini for like a human you have hii inakaa hivi. Halafu kuna ingine inakaa hivi. Unaona ziko tofauti. Okay? So you say homodons possess the same type of teeth, while heterodons possess different types or kinds of teeth. The next question is,
A certain animal has no incisors, no canines, 6 molars and 6 molars in the upper jaw. In the lower jaw there are 6 incisors, 2 canines, 6 premolars and 6 molars. Write down the dental formula of this mammal.
so read again, it has
It has no incisors.
kidogo. So, incisors means
0.
0,
No canines.
canines 0,
6 premolars,
premolars 6,
6 molars
6.
in the upper jaw.
Oh, this is in the upper jaw.
Yeah.
Upper jaw. Then
In the lower jaw
lower jaw.
there are 6 incisors,
6 incisors,
2 canines,
2 canines,
6 premolars
6 premolars,
and 6 molars.
6 molars. So you are taught to write the dental formula. So we know when you are given the dental formula, you are supposed to write one side kwenda huku. So one side kwenda the other. So that utakuja useme incisors. Kama iko na incisors, the lower jaw iko na 6, inamaanisha 3 pande hii, 3 pande ingine. Sawa? So incisors ni 0/3. Then unaweka comma. Next ni canines 0/1. Sawa. Then next, nimeandika place. Kuna space so in caesar equals to 0 over 3, comma c 0 over 1, then comma 3 more 3 hapo more. Then nikanga pm pm. Yes. Then m is equals to 3 over 3. This one is what we are expected to write. The dental formula. The next question is what is the name?
What is the mode of nutrition of this animal?
Look at that. What is the mode of nutrition? It doesn't have incisors on the upper jaw. It does not have incisors and canines. Na meno yake ni ngapi? Anaweza count. That's 2. So that animal is canine, herbivore, or omnivorous. So first it is not omnivorous. So first it's not omnivorous, it's not a human being. Haya. So umebaki na herbivorous ama carnivorous. Carnivorous siwezi kosa meno hapa juu. Hata mbwa unaijua. Unaijua? Wangalia mbwa. So these are herbivore. Inakuanga na kiboni juu. Oh, not herbivore, sorry. Herbivorous. Umeambiwa nini?
Mode of nutrition.
Unaandika herbivorous. If you write herbivore, unaandika herbivorous. Herbivorous. If you write a herbivore, unaandika reject herbivore. So you write herbivorous. It's herbivorous and so you see biology, you see your haina grace kama chemistry, you write the right thing but naona spelling is wrong, I think. Sasa biology utaanguka ukiandika hapo. The next question 8. The reaction is presented by the equation below. So, hydrogen peroxide when you have from hydrogen peroxide inaandikwa aje? Oxygen. Even it is in form 1, oxygen plus water. Lab preparation of oxygen. You use hydrogen peroxide. I think you put a catalyst. Hiyo ni manganese composite kama inakuwa hii. So hydrogen peroxide inakuwa H2O2. O2 plus H2O. Then you can balance. We say 2. Then 2. It is balanced. Oxygen 4, oxygen 4, hydrogen 4, hydrogen 4. Hi. Name enzyme Z. Don't say manganese. It's an enzyme. We are not talking about. We are not talking about chemistry. Chemistry, you would be asked about the catalyst. So, what is enzyme Z? That Oh, haijaandikwa. So hapo unaweza andika enzyme Z. Enzyme Z. So you are being asked about enzyme Z. What is it? Tumesema in chemistry we call it a catalyst, then you identify. So this enzyme is a catalyst. 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 Enzyme catalyst. So, the next question. Name an organ in human body where this reaction occurs. Catalyst. It's a catalyst. It catalyzes hydrogen peroxide into water and oxygen. Write an organ in the human body where this reaction occurs. This occurs in the liver. This occurs in the liver. Then wanauliza state the biological importance of the reaction. Biological importance of the reaction. It occurs. Importance means breakdown of hydrogen peroxide into harmless. Breakdown of hydrogen peroxide into harmless. Into harmless water and oxygen. So hydrogen peroxide can be harmful. So it is broken down in the liver into harmless oxygen and water. Harmless products, water and oxygen. Number 9. Inaweza exist. You know, hydrogen peroxide is ni water and you can excess oxygen. It can be present in your body with excess oxygen. And in exists. Iko kwa blood ikipita kwa liver inakuwa broken down into water and oxygen. So tulisema kwa excretion kwa form 2 that there are many functions of the liver. Over 500 ama 5,000. There are so many that we only learn a few. So hiyo ni function moja. Ukiulizwa kwenye tu unasoma biology about functions of the liver, you can say breaking down of hydrogen peroxide into water. State how So the adaptation is thus, it has numerous chloroplasts. So that's the adaptation. Chloroplast kazi yake ni photosynthesis. Palisade cells ziko kwa leaves. So has numerous chloroplasts which carry out photosynthesis. Adaptation. The next question.
As an acrosome that contains lactic acid and that is the membrane that closes the sperm.
It has an acrosome. That the acrosome does what?
Release the egg.
So this digest egg membrane, ama ovum, the membrane of the ovum, membrane of the ovum during fertilization. During fertilization. Remember, fertilization is the fusion of the nucleus of the two cells, the ovum and the sperm cell. Another adaptation of the sperm cell. Another adaptation. What allows it to swim to meet the ovum? The tail, which has a long tail. A long tail. The other one? It has a large nucleus. Another one, adaptation of large nucleus. The role of the nucleus. The role of the nucleus, penetrate nucleus. The head is penetrate, but the nucleus, kazi ya nucleus mpaka kwa plant cells inakuwa carry the genetic material, DNA. Nucleus. What's the nucleus? The function of the nucleus of a cell. You have your names confirmed. One role inakuanga to control the functions of the cell. 2, to carry genetic materials. DNA. Ni kweli? Mtaisoma tena kwa mfano. DNA inakuwa kwa nucleus. DNA. So has a large nucleus full of DNA. Ama inakuwa na chromatin material. The last adaptation, by the way, kaulizo sasa 4 adaptations. You see, you should write all those. Ya kwanza niyo has a tail, ingine has acrosome, then large nucleus. Ya mwisho has many mitochondria. It's a cell. Sperm cell is a cell, so has many mitochondria. So tulizo hapo ya mitochondria ziko nyingi, muscle cell and sperm cell. You see, that's a point you should write, that my numerous mitochondria. You can also note somewhere that mitochondria are found in abundance. Are found in abundance in muscle cells, and sperm cells. So nikiuliza wakati name two human cells where mitochondria is found in abundance. It doesn't mean kwingine hakuna ziko, lakini kwa sperm cells ziko nyingi, and the function is to the sperm cells require energy to swim to the ovum, so the mitochondria produce energy, synthesize. So energy in form of it is high. Number 10. Wisdom. You are the next person to somewhere swallow.
State the function of the body, each of the following organs.
Lysosomes. So functions of the following organelles. Nimesema lysosomes ama ribosomes. The function? are sexual protein synthesis. Umeandika hiyo ribosomes size for protein synthesis. The size proteins. So ribosomes sites of protein synthesis. Haya, Golgi apparatus. Golgi apparatus. Functions of Golgi apparatus. Hiyo nafaa nawapasha kweli. That's all you know.
Package and transport proteins as secretions.
Package plus transport.
Glycoproteins as secretions.
Glycoproteins. And secretions.
As.
Plus.
As.
S. Yeah, S. S secretions. Golgi apparatus. Packaging and transport of glycoproteins as secretions. The next is explain how the following factors affect the rate of photosynthesis. Concentration of CO2. CO2 concentration, how does it affect the rate of photosynthesis? Does photosynthesis require CO2? So, concentration itafanya nini? Increase in the increase in CO2 concentration, it increases rate of photosynthesis. So, because CO2 is a component of photosynthesis. For carbon fixation in the dark stage. Light stage, we split water molecule to hydrogen and oxygen. Hydrogen ion and oxygen gas. Then that hydrogen ion is combined with CO2 to form glucose. I wrote for you the other time here hydrogen ion plus CO2. Alafu ndio glucose itakuwa CH of itakuwa na hydrogen itakuwa na carbon itakuwa na B6 12 6. Haya. So you say rate of photosynthesis increase as CO2 concentration increases. Rate of photosynthesis increases as CO2 concentration increases up to optimum level. Rate of photosynthesis increase as CO2 concentration increases up to optimum level, and vice versa. Rate of photosynthesis increases, the CO2 concentration increases up to optimum level, and vice versa. The other thing, wamesema light intensity. Does photosynthesis require light? Yes? Kwa agriculture kuna light intensity, light wavelength, vitu kama hizo. Wisdom ya doing agriculture? Ningewafunza agri. Nataka kufunza wisdom. Haya. So since photosynthesis requires light, you just say increase in light intensity will increase photosynthesis. Rate of photosynthesis increases as light intensity increases up to optimum level. Haitaendelea ikiongezeka kiwango endelea ikiongeza light, imaanisha photosynthesis itaendelea tu. But light intensity, photosynthesis, the rate of photosynthesis increases as light intensity increases up to optimum level, and vice versa. Number 13. Number 13. Is it number 13? Number 12. Explain how the following factors in 11. 12. Study the figure below which shows a type of epithelial tissue. State the name of structure A. State the name of structure A. Inaitwa nini epithelial tissue? Meaning epithelial tissue means the last part of your skin, even in your gut, in your digestive, alimentary canal, the outer skin inaitwa epithelial tissue, sio? What do you have? So A, I see you are writing. So, macro villi. Ikiwa moja ni villas, nyingi microvilli. Moja ni microvilli. Haya, give an example in humans where this epithelium is found. Where is this epithelium found? Organ. It is found where? So it's found in the ileum. So, you can also say ciliated epithelium. Confirm that diagram. That's the exercise book. Kama hivyo nifungue kuna hii dada wale walikuwa wamechora ilikuwa exact. Hiyo ni nimechora sasa. This is not a microvilli. Unaona vile inakaa ni katu nini? Hamna hiyo ni this is not a microvilli. Nimeangalia vizuri. Kwa naona ni kama ya mdomo. It's not a microvilli. Ciliated epithelium. Cilia ni kwa kunyonya. Ciliated epithelium. Ciliated epithelium. Ciliated epithelium. Where is it found? In the trachea. Ama unaitanga vipi? Trachea. Yeah. Sorry for that. Only the trachea. So that is question 13. So you can also say trachea ama naso. Ya nose, inaitwa naso epithelium. So unapata cell iko na nywele inaitwa ciliated. Ndio nywele inaitwa cilia. The difference between my diagram na microvilli, utaona microvilli inakaa hivyo. Halafu you'll be able to see, lakini ukiona inakaa hivi. Halafu nywele. This one. Hii ni ciliated. Ni cilia ile. Ciliated epithelium. Haya. Question. Kuna kitu mpaja leo? Hakuna kitu mpaja leo. Haya. I want to observe time because I have a task I want to give you. The other day kumekuja nikapatia watu task and it was so nice. Wakuja wakatu present here. Here number 14. 14 nasema tu. Is it 14? Nimeruka. Number 13, the diagram below represents part of a human digestive system. Name parts labeled L and M. The digestive system, part L and M. M ni she has a question. Sijui mtamove penye yako. Naona ameandika all the questions. You have written all the questions. Have you copied the questions? Umeandika two answers? Part L and then nikiwapea mnisomea swali ya mnisomea. Part L ni part L ni duodenum na part hiyo ingine duodenum na hiyo ingine pancreas. K. Juu next time tutaulizwa K. L ni pancreas. Sorry, K. K ni gallbladder. Hey, kani mfanishwa hivyo? Nimeona msamani. I hope mnaelewa. Kuna kitu mtu hajashika? Halafu kuna kitu hajashika? Nikupatie kati tena umechapa tu yote 100, si ndio? Very good. Some Form 1 students wanted to collect the following animals for study in the laboratory. Flying, state the suitable apparatus they should use for this collection. We have flying insects, crawling, stinging animals, other insects, and small animals from tree barks. Flying insects, hiyo ndio karali. Sitting nets. So for flying insects, you use a sweeping net. Then crawling, stinging animals, you use a crawling, stinging animals kama siafu, maybe a centipede. Centipede is poisonous. Millipede and a centipede, there is one that is poisonous. But do they crawl? Do they crawl? Ama tuseme hiyo inapanga na last thing in Kikuyu we call it kekunyamba. Inapanga hivi tumeguu hapa, tumeguu halafu inafanya tumanya twingi. Unaelewa? Hii kuna manyoya nyingi, ni mdudu tu lakini hiyo ni stinging. Ukimweka hapa utaanza kujikuna. So what suitable method can you use to collect that? Pair of forceps. Pair of forceps. Forceps and beds. Forceps. Small animals from tree barks. Use a putter. Use a putter. Kwani kwa dakika kwa sababu a student estimated that the diameter of a field of view is 2.8 millimeters. So estimate that the field of view is 2.8 millimeters. The diameter was occupied by 4 onion cells. So, estimate the micrometer, the diameter of one cell, and show your working. Hiyo swali si ni anasari 2 marks. The diameter of one, kupima hapa na umepewa hii yote ikiwa 4 ni ngapi? Unapima moja. Hii inafaa kuwa hapa ma. But show your method. You don't just write your answer. So how do you solve this? Diameter of one cell. Tulisoma na nini kara? Diameter of one cell, what do you do? The difference between? Jana soma your lips. Stop chewing. Hata nini? Haushiki swali. Ninakuuliza. Diameter of field of view. Diameter? Useme what you know. Is it go to number of cells? Counted never? No. Maana ukisema tunakucorrect, ukinyamaza how do we correct you? You are saying that the diameter of one cell, or width of one cell, is the number of cells counted that they are formed, divided by the diameter of field of view. Hiyo ina make sense? Siangalie tu kwanza useme hivi hapa nikaa kusema kutoka hapa mpaka hapa kumewekwa tiles 4. This wall kumewekwa tiles 4 na zimejaa. Unaulizwa size ya tile moja. Ni hiyo length divided by the number of tiles. Sindio? So diameter or width, diameter or width of a cell, diameter width of cell equals to diameter of field of view diameter of field of view divided by the number of cells. Number of cells counted. So that the field of view is 2.88. Cells counted 4. So kila moja inachukua 1 point, inachukua 0.72 millimeters. Which one? 2.8. Sorry, 2.8. This is 0.7. Sawa, millimeters. Wamesema ni micrometers. Estimation in micrometers. Oh, so then 1 millimeter forms how many micrometers? 1,000 micrometers. Micrometers, it's like a U. Then hapo unasema uko na 0.7 millimeters. Itakuja ngapi? We just multiply. Your answer is 700 micrometers. Micrometers. Umeelewa hiyo? Haya, usikose tena. Na usikram, nimekuonyesha how you do it. Just think that you have a wall that has 4 cells and 4 tiles. Then that length of that wall iko hivi, inatoshana hivi 2.8. Alafu kuna 4 tiles. What is the length of 1 tile? And somehow I think sometimes we fail because we think that what we learn is not applicable in kitu nje. But the moment you start thinking about it logically, it becomes very easy. Number 16. The diagram below represents the structure found in the walls of helium. Name parts. Identify the structures shown in the diagram. The structure is biologist. Helium. Is not the helium. Is it helium? Villus. It is found in the helium, but it's not the helium. Kuna tofauti ya helium, hiyo ni the villus. Na ni moja usiandike villus. Haya. Name parts. Villus inaandikwa hivi. Name. So that's the villus, the diagram. Name parts labeled S, T, and U, and L. Karibu our students. Acha tumalizia akawasalimia. Sawa. So the next question, tunasoma parts labeled S, T, and L. So you have S. You can see S, T, and L. What is it? S inaitwaje? You have your book. Uko na kitabu au tusome kama sasa. S is the epithelium. What is cell? No, this is the oh, yeah, in the cell. So you can say epithelium cell, and the epithelium, the structure. Ndio kidogo. Haya. T. T ni nini? From your notes. T ni nini? Lactin. So T is lactin. T is lactin. Then you have L. L is okay. Capillary is a blood vessel. Blood capillaries or blood vessel. So it's blood capillaries. To be specific, blood capillary. So this is L. Then we have the next question of functions. Vincent, I told you ukikuja una distract the other class. You should be waiting for your class. You are laughing wengine wakisoma. So name the products which are absorbed into L and T. Products that are absorbed into L and T. L is L. So the blood capillary and the capillary. Now what is absorbed in the lactin? So what type of food is absorbed in the blood capillaries? The first thing you know about glucose, you know. What else? Proteins. So you say glucose, amino acids. Then the other one is fatty acids and glycerol. So for your lactin, in the lactin, there is the fatty acids and the glycerol. Then in the blood capillaries, there is sodium glucose, then amino acids. So what are amino acids? What are amino acids? Yes. What is amino acids? So they are being absorbed in the small intestine. So it is part of food. It has been broken down. What type of food is it? Proteins, carbohydrates, fats and oils, minerals, fats. Amino acids are not fats; they are proteins. The smallest function, not function. The smallest units, they are proteins. When you digest them, they become fatty. They become amino acids. These fatty acids and glycerol, fats and oils. Fats and oils. Sawa. Amino acids ni protein na glucose ni starch ama carbohydrate. Sawa. Haya. So mineral salts kwa zinakuwa absorbed wapi? Kwa nini hatujaweka hapo? Ama water? Where is water absorbed in the alimentary canal? In the colon. Colon, your large intestine. Oh, yeah, in the colon or the large intestine, alongside with the mineral salts. So, kina vitamins. When you eat that food, it's absorbed alongside water. The next question, I don't have all the questions. State how the above function is adapted to its function. Adaptation to its function. So how is the villus adapted to its function? So first it was supplied with the many blood capillaries. Na kazi yake ni absorption. So supplied with blood capillaries to transport absorbed products of digestion. Adaptation of the villus supplied with blood capillaries for transport. Then, to transport absorbed products of digestion. Then, kuna presence of lacteals or lactil to transport fatty acids and glycerol. Then another adaptation. Adaptation ingine, presence of lactil to transport fatty acids and glycerol. That adaptation, the third adaptation lined with the lean epithelium for faster absorption of products of digestion. When we talk about diffusion, we talk about so lined with lean epithelium, lean epithelium to transport products of digestion. Lean epithelium to transport products of digestion. So, lean lined with thin epithelium for faster absorption of products of digestion. Why? Because we want the rate of diffusion equal faster. The epithelium cell inakuwa thin. When the rate of diffusion, the width of the membrane, the width is. Kwa hiyo kuna swali nimeuliza somewhere, I think ni hour from 3. The width of the membrane affects the rate of digestion because of the distance the products are required to diffuse through. So, diffusion can also occur through a membrane. Tumeona hiyo? The first one. The other question is, explain why plant cells do not burst when immersed in distilled water. That one is a repeat question. Nilikuwa nimewauliza. Why? Let me hear the answer from Kishi. Presence of the cell wall. Therefore, if you have a distilled water, yes, it will absorb water through osmosis, but when it becomes turgid, the cell wall will prevent or resist further expansion, therefore prevent further absorption of water. So, that one is it. Number 11, the last question. Yes. The diagram below represents a setup that was used to investigate a certain process in plants. So there is light, there are some leaves, LOD, and gas. So what do you think is being investigated? What do you think is being investigated? So they are investigating the gas present. Which gas? but it's a certain process in a plant. Was used to investigate a certain process in a plant, so you need to put a process. Maybe to help you either photosynthesis or transpiration. Do you think it is photosynthesis? Photosynthesis. Now, how would you know photosynthesis has occurred? How would you know that photosynthesis has occurred? Because of presence of light. So, without light, kama hakuna light, we are not testing photosynthesis. So photosynthesis will take in, will produce oxygen as a byproduct, and then oxygen will be collected. So it is photosynthesis. Then gas B. Gas B. Inafaa kwa oxygen ama CO2? CO2. Why? Plants give out CO2. You give out CO2, but plants give out.
No, my thinking was they require CO2.
They will require CO2. Oh, this one will produce. Unaona kuna bubbles. Haujaona the others. Hawakuona ukiweka. Hawakuona wakiweka, umeona the result. So LOD is a sea plant. Maybe hata inakuanga submerged. Then we have other than factors shown, state 2 factors that would affect the process known in A above. Other than the other factors shown, state 2 factors that would affect the process shown in A above. Other than the factors above. Kwani kuna factors umetaja jamani? Other than light. State 2 other factors that affect the process. So ni kama light intensity, tulikuwa tumesema hapa factors that affect the rate of photosynthesis. One was light intensity, the other one was CO2 concentration. The other is, so kuna CO2 concentration. Temperature, amount of chlorophyll. So any two. So then, as I told you, study questions. B is study to A. So if you fail A, you don't know it is photosynthesis, you cannot get the factors that affect photosynthesis. Uandike transpiration, then uandike zikuwe zina share factors. If they are sharing factors like temperature, hautapata. Okay. So if you fail A, you fail B. A is photosynthesis. Set the process that was being investigated there. A process in plants. Photosynthesis. They were investigating photosynthesis. You just state photosynthesis. Because you are told to state, not to explain photosynthesis. The last question, applying 2 roles of active transport in human beings. States applying 2 roles of active transport in human beings. One role is tuseme active transport inatumika wapi in the human body? We said with you akisoma na hawa mpaka plants. So absorption of food. Kwanza unatafuta tu kwanza penye diffusion iko. Sawa. So now food will diffuse. Food will diffuse ikiwa hapa. So this is food will diffuse. because inatoka from the region of high concentration to low concentration. What about as it moves along the alimentary canal, then after food imekuwa absorbed, hapa sasa kuna food nyingi tukaandia. So the rest will be absorbed through active transport from low to high. So absorption of food in the ileum, that is 1. 2, reabsorption of useful substances in the kidney tubules into the bloodstream. Reabsorption of useful substances in the kidney tubules into the bloodstream. Ingine, movement of waste products from the body into the bloodstream during excretion. Movement of waste products from the body into the bloodstream, ama excretion of waste from body cells into the bloodstream. Movement of waste products from the body, ama body cells into the bloodstream during excretion of waste products. Hiyo umeandika, those are 3. So any 2. Kuna swali on that part? Do you have any question? Na nini wazuri, hamna swali. Mko poa? Okay. Now we have come to the end of that one class. Your part.